some more about genetics. Last time I looked at faults in genes, looking at mutations and how we can look at some of those to find out what's going on. And we looked at the lac operon. And I want to sort of carry on from there, and I want to move on and look at things like cyclic AMP. I want to look at post-transcriptional and translational control, and then move on to epigenetics. So let's have a little look at some of this. If you're good as well, we'll have a look at more about PCR machines, because I want to look at sort of how some of those work, because it's quite relevant with what's going on with coronavirus at the moment. So, how do things affect genes? There's a beautiful experiment that I watched a while ago now, and that's where they took a butterfly. They took a caterpillar, it made a pupa, and you then leave the pupa, and basically the cells all turn into sort of mush. They reorganize themselves, and out comes a butterfly. And someone tried an interesting experiment. What they did, they waited till the caterpillar had liquefied, they opened up the pupa, they mixed up all the cells and put it back. And a few weeks later, out came a beautiful butterfly. It didn't make any difference that they'd moved all the cells around. They sort of knew how to get to the right place. They knew how to switch on and become the right sort of genes for the right sort of cells in the right sort of place. And that's quite interesting, how chemicals affect the plan of the body of an insect and what goes on. Let's start by having a look at the regulation of transcription and translation and see what we can do with this. I'm going to have a look at the effect of estrogen, because it's a nuisance of a chemical. Right, let's get going. I'm just sorting myself out. Zoom in. Right, so let's have a look at a little bit on the effect of estrogen. So what I've got is a cell membrane, and this is going to take an awful long time to draw. So we're going to have a very short cell membrane. There we are. And this is our outside of the cell, and we'll have the inside of the cell. And we have basic control. And so out here comes an estrogen molecule. And this estrogen molecule, because of its nature, can diffuse through this membrane, and so this enters the cell. Once inside, it can bind to some sort of protein, and what I'm going to do is I'm just going to draw my protein in here, and I think you can remember that's the estrogen. And what we've got here is something that will perhaps bind to DNA. So this is basically something which we will call a transcriptional factor. So the estrogen here is going to sort of bind to this protein, and as it does so, basically it's going to change the shape of this particular part of the molecule. So when the estrogen binds, to the protein, then it's going to change the shape here of the DNA binding site. Having changed the shape of this DNA binding site, this can then enter the nuclear membrane, and as it does so, then this particular molecule can now bind to a piece of DNA, yeah, it doesn't look like a helix, I know, it's about the best I can draw here, and this will somehow affix itself to a piece of the DNA, and it will go ahead and produce mRNA. The alternative of this system is, of course, that estrogen does not go through this membrane. If it doesn't go through the membrane, then this protein won't be changed, and so therefore this, although it may go through the nuclear membrane, it won't bind to the DNA, so therefore a gene won't be copied, the mRNA won't be made. So estrogen here is quite an interesting substance. It's a hormone, and it basically is causing a gene to be expressed or not, I suppose. And this means that we can switch on a gene from outside, and this basically is really useful. Can we do it with other things? Yeah, we find that there are a whole variety of different chemicals that are affected in this way. We all have hemoglobin in our body, but when you're conceived and you start having and making hemoglobin, your hemoglobin needs to be different to that of adult hemoglobin. Now, why is that? Well, let's look at fetal hemoglobin. In adult hemoglobin, we've got our lungs. These are full of air, and it's got air at sort of oxygen here at quite a high partial pressure. Let's say it's got it at about 16 to 20%. The body there is oxygen. When the oxygen goes around in the blood, it arrives at the placenta, and at the placenta we've got oxygen now at a lower concentration. Let's say the concentration here is now going to be 12%. So at this sort of percentage, the hemoglobin is going to take up the oxygen, and at 12%, hemoglobin is going to release the oxygen. Now, if the baby, the fetus here, has the same hemoglobin, then at 12%, it's not going to pick it up. it would actually be trying to release oxygen. So the hemoglobin is modified so that at 12% the fetal hemoglobin is going to pick up the oxygen, take it around to the body, and it's going to dump this perhaps at 8 to 10%. So the baby's going to be working on a lower concentration than probably the adult is, because it's a body designed to run off another body. When the baby's born, then this fetal hemoglobin is now no longer required, and a better hemoglobin is required, and the lungs opening up will gradually enable this fetal hemoglobin not to be needed anymore. And it's not needed anymore because a chemical messenger isn't being made. So we work out that the fetal hemoglobin's got a greater affinity for oxygen than adult hemoglobin, and we've got different genes controlling this, and it depends on which of these genes are expressed at the different times to what's going to happen. So all this causes the expression of different genes. Now, one of the courses controls how some of this works in the body, and I'll outline some of that now. Chemicals to produce the RNA are really RNA polymerase. RNA polymerase only needs basically a slow rate of transcription, and if we increase this or decrease this, then things can be changed. We looked at the lac operon last time, and we found that we could basically put some inhibitor in here. So we've got our standard binding site, and then we have, if you like, an inhibitor site, and then we have the gene. And if we can put something here, then the RNA polymerase can't go on. Now, that's the lac operon, and was used to try and explain and understand how all this works. Basically, we've also got another little thing in the system, and this is called cyclic AMP or CAMP. AMP, adenosine monophosphate, which will make later ADP and ATP. It's a very much used molecule, and in this form it's just cyclic. Now, this cyclic AMP can basically bind to one of these proteins, part of this RNA polymerase. So we can find that perhaps if we look at something like the transport of glucose in something like E. coli, it will decrease, the transport of glucose will decrease the levels here of cyclic AMP, which will then reduce the transcription of all the genes responsible for the metabolism of things like the lactose. So if both glucose and lactose are present, then there's still going to be glucose, and that's the preferred respiratory substrate, and that one's going to be metabolized. If there isn't, then we have a need for more lactose to be broken down into glucose and galactose. Something I hinted about last time was we looked at our piece of DNA. So let's have a look at a gene here, and this is just a gene, it's a piece of DNA, and this DNA can be thought of as having different bits, and we're going to call these different bits an exon and an intron. Now, what happens here? Well, only in eukaryotic cells does this happen. We get this copied into a piece of mRNA. So the whole gene gets copied, and this makes something called pre-mRNA. This pre-mRNA needs to be modified to form the proper mRNA that's going to be used. And what we have is these bits of code, and let's have this here as our introns. Basically, these need to be excised from the mRNA so that these other bits can then be joined together to make the mRNA, and these then fuse together, and we find that at either end we've got other bits and pieces. One end we have something called a cap, and the other end we have something we'll call here a tail. I've drawn them funny shapes, but in fact they aren't significant, they're just so you can identify them. A cap, which is a modified nucleotide, is added to this 5' end, and the tail, which is a long chain of adenosine nucleotides, is added to the 3' end. Both these help, if you like, to stabilize the mRNA, and this basically delays the degradation of this in the cytoplasm. The cap also aids binding the mRNA to the ribosomes, and we've got this splicing occurring here where the RNA has been cut at specific points. The introns, which are sort of non-coding pieces of DNA, are removed, and the exons, which are the coding pieces of the DNA, are joined together. And this all occurs within the nucleus. The nucleotide sequence here of some mRNA molecules can also be changed here with some base addition, base deletions, or substitutions. And these can have the same effect as, if you like, point mutations. And it's interesting how this RNA, when it's forming like this, can be edited and modified, and it's done sometimes by these extra factors. So one of the things that's not really well covered when I look at some of this is the fact that, left to its own devices, mRNA, basically it degrades outside the nucleus, and basically it just disappears. It's broken down into its constituent parts, which can be reused over and over again. This is useful because a cell doesn't want to keep producing this protein all the time. So these have a sort of fixed life. By putting on a cap and a tail, this increases the life of these pieces of mRNA, and that means that they can last significantly longer. Right, so that gives an idea on hopefully some of that. Now what we're going to try and do is look at more about how some of that works, and we're going to look at something called the epigenome. So let's have a look at how some of this works. And I'm not very good at drawing this, so you're going to have to bear this in mind with me. Right, okay, so first of all, what is the role of the epigenome? And the epigenome basically influences which genes can be just transcribed. So epigenetics, it's basically controlling which genes in a cell are basically able to be transcribed. So if I've got my piece of DNA, and the drawing doesn't get any better, what we can do is we can attach different molecules. We can attach a methyl group, or we can attach an ethyl group. Methylation, or we would call this one an acetyl group, an acetylation. So even though we know it's an ethyl group, it goes by its old name in biology. So the attachment of this methyl group, which is a CH3 group, to the gene, and that usually binds to a cytosine, prevents the transcription to the mRNA. Now that's fairly obvious to think that if we've got something blocking one of these bases, then it's certainly not going to be transcribed. The modification also means that proteins can join on, and the proteins are called histones, and these are found only in eukaryotic cells. That's not absolutely strictly true, but it'll do from where we go at the moment. Right, okay. So what can we discover with some of this? Well, we know that DNA is wrapped around proteins called histones, and if I were to draw a couple of histones, the DNA, if you like, goes round the histone like this, and probably we'll have another histone round there, and I can have another histone round here, and it goes round in this sort of shape, basically forming sort of these little loops. Now we know if the DNA is like this, then basically we've got no transcription. Because how can the mRNA polymerase get in if it's all wrapped round this? So we've got a basic form of control, that if we are a particular type of cell, and cell A is going to produce protein A, and we've got cell B, and this is going to produce protein B, and these proteins give an idea of what this cell is going to be and what it's going to do. If we take our control of this, we've got some sort of probable outside control saying where these particular cells are in relationship to other cells. So this cell knows where it is because it's next to or near to other cells, and there's some sort of concentration gradients of some particular chemicals. And if we know that it's here, then we have our piece of DNA that goes around there, a clear bit, and then it's covered in histones, and this is the site for protein A, and protein B is locked away, whereas in this cell protein A is locked away in here, and protein B is here available, and it's simply whether these different bits of DNA are opened up, whether they are hidden by these histones. So this epigenome is determining the shape of this DNA, and it keeps genes that are inactive tightly packed and ensures that they basically can't be read. In other words, they're switched off. And this is given a name, and this is called epigenetic silencing. By contrast, if it's unwrapped, then the DNA is exposed and it can easily be transcribed. Therefore, it's switched on. We know that DNA code is fixed. The epigenome, however, is flexible. And so we have cells that can do particular jobs. When you're born, when you're conceived, to be more precise, then your cells can produce every different type of possible gene, and these are, I'm not going to spell it, am I? Totipotent. Later, these cells don't need to make every bit, so they don't need to make things like placenta, umbilical cord, and various other things in a human, and so these cells become pluripotent. And those cells, the only difference between them is that they've got epigenetic silencing on some of those genes. All the genetic material is still there in both cells, but some places they're turned off. And later, the cells with the same DNA, become specialized. And then they can only produce the genes that are required for that particular cell to use. So the epigenome of a cell is basically the accumulation of a whole series of signals it's received in its lifetime. And we can think of this epigenome then as perhaps a cellular membrane. In early development, signals come within the cells of the fetus. They're going to come from the nutrition provided by the mother. After birth, various signals are going to happen. Hormones maybe are going to be produced, and they're going to cause the epigenome to activate or inhibit certain sorts of genes. Let's go and have a look at this epigenome in a bit more detail. So we've got this DNA combined with histones, and this is given a particular name. It's called chromatin. So where the association of histones and DNA is weak, we've got less basically well packed, less condensed, then the DNA can be accessible to various transcriptional factors which can produce the mRNA. Where this association is strongest, the DNA histone complex is more condensely packed, and therefore this DNA is not accessible by any of the transcriptional factors, which means that mRNA can't be made. This chromatin is basically condensed, and we see that when we're looking at cell replication. During cell replication, nothing is being made. Well, not fleeced from the DNA. Any mRNA that's already been made, yeah, that can carry on doing its job. But the DNA can't be read when you can basically see these genes in the form of the chromosomes, when you can see the chromosomes. Right, so if we do that, then it's produced, this condensation is produced by decreasing the acetylation of the histones or by the methylation of DNA. So let's look at this. So we've got two choices here. We can decrease acetylation. Basically, acetylation is a process by which an acetyl group, which is CH3, CH2, here is transferred to some sort of molecule of some sort. In this case, this group here is normally added by our old friend acetyl-CoA from glycolysis and various other bits and pieces. The link reaction. Deacetylation is the reverse reaction where this acetyl group is removed from a molecule. Decreasing the acetylation increases basically the positive charges on the histones and therefore their attraction to the phosphate groups of the DNA. And we know that like charges repel, and so they're being pushed away. We can increase methylation. Methylation, the CH3 group, and that is added to cytosine bases of DNA, and that's going to inhibit transcription. It's going to prevent the binding of the DNA, so it's going to prevent binding of transcriptional factors. Let's get it right. To the DNA. It's also going to attract proteins, and when it does that, these proteins that will condense on the DNA histone complex, therefore it will induce deacylation of the histones, and that's going to make the DNA inaccessible to transcriptional factors. So what do you look like? Yeah, I didn't show you that bit. Right, so what do you look like? Well, you look like the genes of your mother and the genes of your father. But in fact, that's not the whole story, because as you develop, your epigenetic or epigenesis is going to also develop. And they've done experiments on rats, and they've shown that female offspring who receive good care when they're young respond better to stress later in life, and themselves they will nurture their offspring better. Female offspring receiving low care nurture their offspring less well. Now, what can control all of that? Well, there's something we meet in humans, and this is called gestational diabetes. Gestational diabetes means that the fetus will be exposed to high concentrations of glucose. This high concentration of glucose causes epigenetic changes in the DNA of the offspring. And that means that in a daughter, there's an increasing likelihood that she would also develop gestational diabetes herself. In the early stages of cell development, then we find that the epigenetic tags can in fact be wiped clean, and there are special processes to try and do this, because if we can wipe out this, then gestational diabetes won't occur. Can we try and treat diseases with some of these then epigenetic therapies? Well, the idea is that we might be able to use some drugs to inhibit some enzymes involved in either histone acylation or methylation. Drugs that do this might be effective. They might be effective especially on things like cancer cells, and that can be quite useful. Right, so we can also look at the effect of basically RNA on some of this stuff and how we can affect sort of cancer. I suppose if I'm going to tell you about that, I need to be a bit better about what cancer is. So let's do this. What is cancer? Well, we know there are two sorts. Yeah, it didn't look like I was spelling that right. We've got benign tumours and we've got malignant. There we go. We've got benign tumours, we've got malignant tumours. Right, now benign tumours, they can grow to a very large size and they grow slowly. But the malignant ones typically grow quickly. The cell nucleus here in benign cells tends to look relatively normal. Whereas here in malignant cells they tend to look a bit different, and there's usually a lot more DNA present that's visible, that's active. In benign tumours the cells often differentiate. And in malignant tumours they are typically unspecialised. The cells here, because of this, stick together, and here they don't stick. And here you can see this is where the problems begin with malignant ones. If the cells don't stick, there they can spread out to all sorts of parts of the body, and they can cause tumours occurring anywhere in the body. These tumours are surrounded by some sort of capsule of dense tissue, so they remain fairly compact. The tumours here are not surrounded by this, and so they grow out finger-like projections. So this has got the finger-like projections, and this is basically encapsulated. This means if you're trying to remove a tumour that's benign, quite often surgeons, rather than using a scalpel, often use fingers, and they can literally pop out some of these benign tumours. But with finger-like projections going into all sorts of other cells, a surgeon here would have to not only cut out the tumour, but would need to try and cut out all these finger-like projections, because leaving just one cell is going to cause it to carry on. And so they've got to start cutting away other tissue as well. So these tend not to be as life-threatening, although they can cause blockages which tend to kill. This one is starting to replace some tissue with other tissue, and this is going to have a big problem. People with malignant tumours are going to have basically systemic effects such as weight loss, fatigue. With benign tumours they tend to be localised, these can easily be removed by surgery, but the malignant ones usually involve some other treatment here as well, and we're looking at, for some of these, radiotherapy or chemotherapy. or both. If you can get this one out, you're relatively safe. Malignant, well, you've got problems. So, where could we go with this? Well, cancer, and this is going to affect cell division. A cancer cell basically is a cell that is going to reproduce. All our cells reproduce, but these reproduce all the time. Somehow this initial mutation that we've got causes uncontrolled mitosis, and basically it's going to grow and grow and grow. The two main types of genes that play a role in cancer are tumour suppressed genes and oncogenes. Oncogenes are mutations basically of proto-oncogenes, and these proto-oncogenes stimulate the cell to divide when growth factors attach to protein receptors in the cell surface membrane. That activates genes and causes the DNA to replicate and the cell to divide. If this proto-oncogene is mutated into an oncogene, it then basically is permanently activated, and basically this means that the cell is going to reproduce again and again and again. Basically a few cancers are caused by this, and if we can try and do something to stop this oncogene, then we might be able to suppress it. There's another type which is called a tumour suppression gene. I don't watch the screen enough to see where I'm writing. Sorry. Tumour suppression genes slow down cell division. They repair mistakes in the DNA. They tell cells when to die, which is called programmed cell death, really. Apotheosis. So they've got the opposite role to a proto-oncogene, and these normal tumour suppressor genes maintain the normal rates of cell division, and so they prevent the formation of tumours. However, if this gene becomes mutated, it's inactivated, it's switched off, then it doesn't stop the cell reproducing, and we now get these cancers starting to form. And there are a few interesting names of some of these genes which you might come across if you do some reading on this, and we come across TP53, and we come across BRCA1 and BRCA2. Enough of that bit. So how do these things link with what I've been talking about with these methylation? Well, we know about methylation. We can find that in these tumour suppressing genes we can get hypermethylation, an enormous amount of methylation. And this leads to the tumour suppression gene being inactivated, and as a result transcription of the promoter regions of the tumour is inhibited, the tumour suppressor gene is silenced, switched off, and it doesn't slow the cell, so we carry on. And this abnormal methylation of the type occurs in this BRCA1 gene, and this is primarily the cause of something like breast cancer. Another form of abnormal methylation could be, instead of hypermethylation, hypomethylation, reduced methylation, and that can occur in oncogenes where it leads to their activation. We started talking about estrogen. Estrogens play a role in regulating the menstrual cycle in women, and it's known that after menopause a woman's risk of developing breast cancer usually increases. It's thought this is due to an increased amount of estrogen. That's a bit of a paradox, isn't it? Because basically the production of estrogens from the ovaries diminishes in menopause. However, the fat cells of the breast cells tend to produce more estrogen after menopause, and these locally produced estrogens trigger the breast cancer in postmenopausal women. And again, once tumours developed, this is going to further increase the production of estrogen, which becomes a sort of a self-fulfilling prophecy. So I've wandered around a topic here, starting looking at estrogen, looking at what it does to cells, how it relates to epigenetics, to how we put histones on, take histones off, and we've looked then back the other way at cells out of control caused by this methylation, acetylation, the removal or addition of these histones, all this epigenetics. Maybe that's given you a few ideas about how things work. I've run out of time to try and do about PCR machine. I think I'll start with that next time, and we'll have a look at see how we can actually do some interesting things with our genes. So until then, take care. If you found this useful, then subscribe. If not, I'll see you same time next week. Take care, stay safe, bye bye.